Hi, my name is Julie and I'm going to present Group 8's investigation on the study of beta-TCP functionalisation of gelatin scaffolds in bone tissue regeneration. The primary objective of this study was to determine if this method is a viable replacement for current bone tissue regeneration methods on the basis of analysing the biological and mechanical capabilities of the scaffolds. The image as seen in the bottom left corner of the poster is representative of the steps completed to carry out this analysis. Essentially, it included the fabrication of beta-TCP functionalised gelatin scaffolds through freeze-drying suspensions that were prepared in the lab. Beta-TCP is very similar to hydroxyapatite, and therefore in the case of bone tissue regeneration, it was expected that this composite material would give the best results. For comparative purposes, GTA cross-linked gelatin scaffolds were also provided, and the mechanical capabilities of the scaffolds were assessed through a uniaxial compression test. This was then followed by a degradation and swelling analysis, and cell porosity and morphology were analysed through scanning electron microscopy. Finally, after a 7-day culture period, MC3T3E1 pre-osteoblast cells were seeded on the scaffolds with an initial seeding density of 1 by 10 to the 6 per scaffold, and the cell viability of each scaffold was quantified through the completion of a DNA hoist assay. The results are in this section above, and figure 1 shows that the effective elastic modulus of beta-TCP functionalised gelatin scaffolds was greater than that of GTA cross-linked gelatin scaffolds at a 5% significance level. Additionally, the swelling tests show that beta-TCP functionalised scaffolds had a higher swelling rate than that of the GTA cross-linked scaffolds, and this variance is a direct result of hydrophobic nature of GTA, may also explain the lack of degradation recorded throughout the study. SEM was used to analyse cell porosity and morphology, and the results from this are seen from figure 3 to 5. A significant amount of pores can be seen on the beta-TCP scaffolds in comparison to the GTA scaffolds. Figure 6 shows a 95% increase in cell viability on the beta-TCP scaffolds in comparison to GTA. In conclusion, beta-TCP functionalisation enhances the physiomechanical and biological properties of gelatin scaffolds. However, degradation was inconclusive in this study, and therefore further investigation into optimising the bioresorbable capabilities of functionalised beta-TCP scaffold is required in order for it to be concluded that this method could potentially replace conventional bone tissue regeneration methods. Finally, I'd like to thank Manus Biggs and his team of lab technicians for their guidance throughout the investigation.